doing the urinary system, and most of us, when we think of the urinary system, think of the role of the kidneys in excretion of waste products, which is correct. Anybody know what the unique element is that's excreted by the urinary system? Right here? Nitrogen, yes. So nitrogen excretion, just the functions of the urinary system, and I'll come back and kind of identify what parts of the urinary system are involved in this. So we have waste excretion, but primarily forms of nitrogen. And so we have urea, which is the most common form. Ammonia and its ions, and creatinine. This is done in a process of filtration by the kidney, but we also see it, this is from beginning to end. So I'm not going to limit it to specific components of the kidney structure. We have pH balance. So the kidneys will secrete or add additional hydrogen ions to the filtrate or reabsorb bicarbonate ions. Electrolyte balance. So the kidney is well known for sodium. Going along with that, of course, is chloride, potassium, calcium ions. There's others, magnesium, phosphate ions, and so on. And this is not a physiology class, so we're not going to go through the cellular processes and pumps and so on of how this occurs, but obviously this happens in the kidney, and I'm going to reference where it takes place. Along with electrolyte balance, we have fluid regulation, some fluid balance. The other day I was walking through the second or third floor of Sequoia, and a gentleman walked by with a container of urine. And physiology classes will have a lab, a urinalysis lab, where you're divided up and you take a drink of high sucrose concentration, high amount of distilled water or salt water. Actually, when I was teaching the course, had a few students approach me and ask, well, can we see the effect of alcohol on urine production? And I said, sure, as long as you don't bring the alcohol to the classroom. So they came to the classroom and did their voiding, well, not in the classroom, but in the bathroom, and then went out to one of our restaurants on campus that serve alcohol, and then came back and did the lab. Because alcohol does have an effect on hormones that regulate fluid balance. In addition, the kidneys are an endocrine organ. They synthesize erythropoietin, which, if you recall, increases red blood cell production. Alrighty, so it has quite a few other roles than just getting rid of nitrogen. Okay? Now, to do all of this, it does so by passing the blood through the kidneys multiple times during the day. Our blood volume is about 5 liters. You make 180 liters of filtrate every day. So obviously we have to have some reabsorption, or we'd be sitting on the toilet hooked up to an IV. We wouldn't be able to go anywhere. We're also having a catheter and a bag down our leg. So one of the reasons why we have those large renal vessels, arteries and veins, is because of this large volume of blood that's moving into and out of the kidneys. All right, so before we move into the kidney structure itself, let's look at the location of the kidney. And we've been talking about structures that are retroperitoneal for the digestive system. We had duodenum, the ascending, descending colon, and the pancreas were structures that were retroperitoneal, meaning they had a peritoneum on their anterior surface, but weren't surrounded by it, not suspended by a mesentery. We look at a transverse section of the abdominal wall. Here we would have our vertebra and abdominal wall muscles. The kidneys are located, of course, on the posterior abdominal surface, and they develop embryonically from the mesoderm, same structures that are forming our muscle, and the peritoneum is formed over them. So we would have, this is looking, look at it like this. All right, so this would be the right side, this would be the left side. The aorta tends to be slightly off-center. And inferior vena cava is more to the center, but also on the right. And we look at the peritoneum, and there's additional adipose tissue over the kidney, so it's not directly associated. The peritoneum is not going to be right on the kidney structure itself. So in a transverse position, this would be the way that we would see it posteriorly. I probably should have done my kidneys in a different color. But posteriorly, if we look at it on a frontal view, we have a couple of muscles that you're familiar with that help to support the kidneys. So if we put I'm going to change the kidneys down to a black color. The right kidney is a little bit lower than the left kidney because of its placement inferior to the liver. And we would have our abdominal vessels here. The renal artery is posterior to the renal vein. And then running along the medial aspect, supporting the kidneys medially, we have a long, slender muscle that comes off of the transverse processes of the lumbar vertebra and crosses the hip joint. Anybody recall what that muscle would be? Functions in flexion. So as major. So if we put our iliac crest here. Remember how I said on the lab exam, structures that we have covered previously that are related back to concepts that we've covered in this unit may appear 5 or 6 of the 54 questions. So I may tag so as major, or the next muscle, which extends from the iliac crest to the lower ribs. So we have just the floating ribs that function to support the upper, no more than the upper third of the kidney. So these would be the vertebral ribs. And what is the muscle that extends from the iliac crest to the lower ribs? One of our deep back muscles? Quadratus lumborum. You can't see that much of the superior portion of the muscle because it's on that portion that would be covered by the part of the diaphragm. It's coming out, so it doesn't look that high in the torso, but it would be present in that position. Now, kidneys are an organ. We have 2 of them. We kind of have a double dose. One kidney is sufficient if it's working properly to provide the filtration that's required by the body. And so kidneys are an organ that can be transplanted from a living donor. Segments of the liver, lobules of the liver can be done as well because the liver will replace its tissue. No other organ will do that. But one kidney can be removed. It used to be that the incisions for that were done in the lateral position, along the lateral flank area to go retroperitoneal and not disturb the deep muscles. Now they're often done with several laparoscopic incisions about 1 inch wide. And then, obviously, the kidney is larger than that. We can't pulverize it to bring it out because then it's of no use. So the incision to bring the kidney out itself can be done just below, above the pubic area, kind of like a bikini incision for a C-section, or it can be done along the midline through the belly button, okay, rather than the large flank incision. Now, the kidneys are surrounded by adipose tissue, so that helps to protect them in location and helps to pack them. I know adipose tissue isn't orange, but it's as close as I have to yellow. And fascia, but once that's disturbed, there's really nothing else to hold it in place. And so jobs that require or provide a lot of bouncing, so truck driving, especially on our roads here in California, horseback riding, doing a lot of horseback riding, and that kind of disturbance can cause one or both kidneys to start to slide down the back of the posterior abdominal wall, which results in kinking of the tube that's going to carry the urine down to the bladder. That's about a long tube. So when a kidney is transplanted, we don't even attempt, or surgeons don't even attempt to put it back up in its original location. It's just going to slide back in. So the kidney will be placed here along the iliac fossa, over the iliacus muscle, and not even attempt to try to reattach it to the renal arteries up here, but either to the aorta or to the common or external iliacs, and nestle it with that bony support of the pelvis. All right, so now let's look at the overall kidney. I'm not sure which came first, the kidney bean or the kidney. Actually, probably the kidney. But I'm going to move over here. We're just going to diagram the growth structures, and then I'm going to come back and fill in the blood supply, and then we'll look at the functional unit of the kidney, which is the nephron. The kidney is shaped very much like a lymph node, much larger. Draining, we have two solutions, three actually, solutions that we talk about in the kidney. Obviously we have the blood. Once the blood is filtered, separation by small particles, we call it the filtrate. And then the filtrate is processed. We remove things from it, we add things to it, and we concentrate it. Once we're done with all of that, we finally call it urine. So the urine is going to leave the kidney from this vessel known as the ureter. And after it's filtered and concentrated, it flows into small, well, not particularly small, but channels that are drained from the outer region. So everything that I've drawn in black here is the minor calyx, two or more minor calyces will join together to form a major calyx. And these structures are all lined with our typical urinary epithelium, which is transitional epithelium. Transitional epithelium, if you recall, is multiple layers. The cells are oriented vertically, so it's a stratified epithelium rather than the horizontal layering of stratified squamous. And the cells on the top are rounded, and occasionally we'll have binucleate. This allows a stretching and a thinning, which is useful in the bladder. So this continues to be the tissue that lines the bladder. The central area that the urine will drain into before it enters the ureter is known as the pelvis. Okay. However, before it enters these structures, it's filtrating. So we're still processing it. Draining into each of the minor calyces is a packaging, a pyramidal. It's actually pyramid shaped, wide at the base, narrow at the tip. When we cut the kidney in a frontal section like this, it looks like a triangle, but in three-dimensional shape, it's a pyramid. And because the structures that form it are all almost in a straight line. So these dip into the minor calyx. Their point where they dip into the minor calyx has multiple openings. So if we were to see one of these coming out at you, it would look like that. And that's where the filtrate drips into the minor calyx, and we now call it urine. Each of these structures looks very striated because the tubes and the blood vessels that we find in them are almost parallel. That gives the striated appearance. So each of these structures is identified as a medullary or renal pyramid, to differentiate it from the medullary pyramids of the brainstem. So the area that contains these medullary pyramids out here is known as the medulla. Okay. So the entire region is the medulla. Remember the lymph nodes had a medulla, that central region, and then the outer portion was the cortex. So from here to here is the cortex. And then, I don't want to make it messy, but the area of blue pyramids is the medulla. Then out in the region out here is where we find our nephron structure. So before we get to that, I want to illustrate the blood supply that is so important. Let's see what I've left open. Okay, the sinus. I'm not sure. We always mention the sinus and then we never do anything else about it. But in this area, or up here, kind of packaging the minor calyces and the pelvis is a depression known as the sinus. The orange that I've drawn in again is adipose tissue. And the best explanation I've come up with for explaining the sinus is important clinically because there are some infections that are in the connective tissue and are in the sinus, and there's others that are bloodborne and are within the nephron or the pelvis. So it's more of a clinical differentiation. But let's say that you wanted to put a pool in your backyard, and you didn't have the time or the money for a custom-built pool, so you're going to buy a fiberglass pool. You dig a great big hole about 2 feet larger than the pool. That's the sinus. Okay, you set the preformed fiberglass pool down inside that hole. That's the pelvis and minor and major calyces. And then you backfill in the space, and that would be the adipose tissue. So the sinus is kind of a depression in the medullary area here that then contains the pelvis and minor and major calyces. All right, so now let's look at the blood supply. And just to keep it from being overcrowded, I'm going to leave the kidney diagram up there, but I'm going to erase the names of structures. Otherwise, it's just going to become too busy to understand. The renal arteries. My best friend in here. Coming into the kidney area. And they're not in the pelvis, they're lying over it, okay? They're not passing into the pelvis itself. And in the area of the pelvis, they form branches known as segmental arteries. And then each of these segmental arteries gives off a branch that passes up between each pyramid. Arteries that pass between each pyramid are known as interlobar arteries. And they reach between the pyramids and out to the cortex. Alright, so segmental arteries here, interlobar arteries between the pyramids. The kidneys actually develop as multiple lobes, and in some animals, you can see this on the outer surface, there's rounded bumps, but in humans, those rounded bumps are flattened. At the base of each pyramid, these interlobar arteries are connected by arching vessels. And so they are known as arcuate arteries. That runs right along the transition from medulla to cortex. Then extending at right angles out into the cortex, we have straight vessels that are known as interlobular instead of interlobar. Or, if it's too confusing to remember which is which, you may call them cortical radiate. They radiate out into the cortex. Your lab manual refers to them as interlobular. So either name is fine. Now, off of those, we have smaller vessels, which are now arterioles. Okay? But rather than add them onto this system, I'm going to take the arcuate artery and redraw it on the next board. So I'm going to take this segment right here, rotate it around, and draw it over here. So here would be an interlobar artery. Here would be another interlobar artery. And then connecting those, we would have the arcuate artery. one of these, and we've drawn it over here. So this would be the medulla. And up here would be the cortex. Let's take a couple of interlobar arteries or cortical radius. Now we reduce the layers of smooth muscle to just one or two. And if you recall, when we do that, what do we call that type of vessel? It only has one or two layers of smooth muscle. It's important in regulating blood pressure. Did I? Arterial. So this is going towards, from after arterial, we lose all the smooth muscle and we have a capillary. Right? The next vessel after a capillary is a venule and then a vein. That's not the case here. So our next structure here is a ball of capillaries. And the blood flow then is going into the ball of capillaries. So this is defined as an afferent arterial. The ball of capillaries are known as glomerular capillaries or just as the noun glomerulus. This is the intimate relationship between blood and the functional unit of the kidney, the nephron. We'll come back and draw it. Now, unlike our normal pattern, instead of having a venule lead, we have another arterial lead. It has smooth muscle again, and a lot of smooth muscle. So it's going away from our capillary, from our glomerulus. And so this is now known as an efferent. Arterial. What would be the advantage of having two arterioles on either side of a capillary bed? Exactly. We can decrease the diameter of the afferent arteriole and drop the flow of blood through the capillary bed and decrease blood pressure. Or we can dilate the afferent arteriole and constrict the efferent, like putting your finger over a hose. It causes blood to back up into the glomerular capillaries and increases pressure in the glomerular capillaries. Okay? And as we'll see in a few minutes, that pressure is what pushes the small particles through the filtration membrane that results in our filtrate. Now, the next set of vessels what happens depends on where we are. So if the glomeruli are up in the outer part of the cortex, the efferent arteriole will lose its smooth muscle again and form a looping bed of capillaries that then empties into an interlobular vein. Which returns the blood flow to an arcuate vein, which returns to an interlobular vein, an interlobar vein. Right? So this looping bed of capillaries is going around our nephron, and it's known as peri, meaning around, tubular, going around the tubes of our nephron unit. Peritubular capillaries. So, just to give you an idea of where we're going, if this is new to you, blood flowing through the glomerular capillaries is filtered across an epithelial filtration membrane. Anything that's small enough to cross that membrane will lead into a structure I haven't drawn yet. The rest of it passes into the efferent arteriole and into the peritubular capillaries. The peritubular capillaries wrap around that nephron and will take back most of the material that was filtered. That's how we go from forming 180 liters of filtrate to excreting 1.8 liters of urine. We reabsorb that material. So we filter it and reabsorb it over and over and over all day and all night long. And the peritubular capillaries are responsible for reabsorbing that material. In addition, they can add drugs to it, hydrogen ions to it, additional potassium to it, and so they can secrete. So that's what the peritubular capillaries are doing. Glomeruli that are close to the pyramids, their efferent arteriole, instead of forming peritubular capillaries, will drop down into the medulla and form a loop of blood vessels that come back up and join into the arcuate vein or into the interlobar vein. So this loop, notice how it's straight? Straight vessels down and straight vessels back up. Remember how I said the medulla has a striated appearance because the structures that are in there are parallel? And so these vessels that go straight down and straight back up are known as the straight vessels, or the vasa recta. So they are found only in the medulla. Peritubular capillaries are found only in the cortex. Structures that we find only in the cortex, vascular-wise, are afferent and efferent arterioles, glomerulus, and the peritubular capillaries. We find the vasa recta only in the medulla. Okay. Now, I'm not including the arcuate and interlobar and interlobular, but as far as these more limited vessels. So that's our blood supply. However, that's not going to be all we need to create our filtrate. We have to take and process the filtrate. So we get to the functional unit of the kidney, which is a nephron. The simplest form of drawing a nephron, all right, I'm just going to draw its simplest form and then I'll draw its more anatomical component. So a schematic of the nephron, what the nephron essentially is a tube of epithelia. It starts out in a large balloon-shaped structure, and this is schematic, all right? Because in actuality, this inner layer is intimately associated with that bed of capillaries. So it's not smooth like this. It's folded all around and wrapping all around the capillaries. This is just a schematic. This portion of the nephron is called a capsule. It's either glomerular capsule, or I still refer to it by its eponym, a Bowman's capsule. Because of its intimate association with the capillaries, the capsule is the site of filtration. We'll come back and to look at its structure a little bit more detailed. So this is where its filtration occurs. Filtration is separation by size. In the kidney, it also includes charge, positive, negative charges. Right? The rest of the kidney is a tube. It's not straight, all right? But if I just look at this, again, this is a schematic. So in the early part of the tube, known as the proximal convoluted tubule, obviously this is not convoluted. This is where about 65% of our water and our salts are reabsorbed. And reabsorption means material leaves the filtrate and goes back to the blood. Filtrate is not blood, it's the filtered material that was taken from the blood. So 65% of what we filtered is reabsorbed. We also will add material back to the blood. So if the blood is too acidic, has too many hydrogen ions, your pH is too low, we will secrete material into the blood. We'll add it. So by the time the filtrate has passed through the proximal convoluted tubule, all the, this is 65% of the salt, but all glucose and all amino acids have been reabsorbed. 65% of the water and so on.
This is just after the proximal?
This is in the proximal, okay? So we've determined the components, all right, of what's going to be in the, we don't add anything new. We may change amounts. We may add additional to what's already there. So we can secrete hydrogen ions and drug metabolites. A lot of drugs and hormones are bound to plasma proteins, which are too big to be filtered, but they'll be added to the filtrate at this point. Then we have a very thin segment of the nephron called the nephron loop. If this nephron loop extends down into the medulla, and that's the only time we care about it, it functions to create a concentration gradient. In other words, the deeper we go, the closer we get to the tip of our papillae, the saltier, if you will, the more highly concentrated the surrounding interstitial fluid is. And that's the role of the nephron loop. It doesn't concentrate the filtrate itself, but it creates an increasingly concentrated gradient. That then brings us to the end of the nephron, which is the distal. Convoluted tubule. The distal convoluted tubule is affected by hormones, primarily aldosterone. And aldosterone functions to increase sodium reabsorption. So the sodium that we reabsorb over here is regulated totally by concentration. We have a lot of sodium in the blood, we reabsorb a lot of sodium. Okay? But let's say that you haven't drunk very much water and your body needs to increase its water, the osmolality, the number of particles dissolved in the fluid is too high. So it releases aldosterone from the adrenal glands, and the body takes in more sodium and water follows it. Okay? So that's a regulated site. This is unregulated. This is regulated. In addition, we have parathyroid hormone that increases calcium reabsorption. And another hormone that I'm going to talk about with the collecting duct. So that's the end of the nephron. Okay. We haven't actually regulated our water uptake, but the next structure that we see with this is not part of the nephron, but is known as the collecting duct. The collecting duct is affected by a hormone known as antidiuretic hormone. This functions to add water channels and now the filtrate is passing back through this highly salty solution, and if we add water channels, well, what happens when you sprinkle sugar on top of strawberries? It osmotically pulls water out of the strawberries. So if you're making strawberry shortcake, all right, you want some strawberry juice to soften up the shortcake, and so you sprinkle sugar on it 20 minutes or so before you serve it. You don't want to do too long, the strawberry's a little gross. All right, so by having these water pores in, that saltier ingredient out here will pull water out of the filtrate, and you'll have a more concentrated urine and a more dilute blood. Water can stay in the blood. Diuresis is creating too much urine, so an antidiuretic hormone is going to return that water to the blood. So in 5 minutes, there's a function of the kidney. But now that's just kind of an overall view of that. Okay. The nephron has a more anatomical structure. Okay. The way it actually looks, and then I'll put it in place on the kidney. Actually, I'm just going to go ahead and do it here. So for the glomeruli that are here up high, we're going to put the capsule, and the inner layer of the capsule is highly wrapped around and associated with each of those capillary walls. So there's our capsule. And then we have our proximal convoluted tubule. I'm just going to write PCT for proximal convoluted tubule. We have our nephron loop. It forms a thin segment here. And then the distal segment comes up between the efferent and afferent arterioles and forms our distal convoluted tubule. And then finally, that enters into our collecting duct. And the collecting duct picks up additional distal convoluted tubules and passes down into the medulla. So when we have a nephron that has a very short loop and it's in the cortex, called a cortical nephron. So this is the descending limb of the nephron loop. Another name is the loop of Henle. This would be the ascending limb of the nephron loop. Distal tubule and distal convoluted tubule. Collecting them. All right. So as you can see, we can add some additional structures that we find only in the cortex. So our capsule. Is only in the cortex. Our proximal convoluted tubule and distal convoluted tubule are only in the cortex. However, our nephron loop, if it's short, remember this is a cortical nephron. So if we have a cortical nephron, the nephron loop is in the cortex. If we have a juxtamedullary nephron, let me just change colors here. Its loop descends down into the deep down into the medulla, comes back up, and we see it again, our distal, our capsule, and everything else is still in the cortex, but now our nephron loop is in the medulla. All right? So the nephron loop can be both. Our collecting duct starts in the cortex, but ends up down at the narrow port of the pyramid. So our collecting duct can also be both. So this is just the general placement of the structures. And you'll see that on models that we have in the lab. And now I want to go back again to the nephron and look at each of these regions and talk about their histology a little bit better more. Okay? So if I may erase this board. Ready? Ullmann's capsule or the glomerular capsule. It is made up of simple squamous epithelium. Remember in the alveolus of the lung, our respiratory membrane, the gases pass through was a sandwich. The simple squamous epithelium of the alveolus, a little bit of connective tissue, and the simple squamous epithelium of the capillary. And that was our respiratory membrane that gases had to diffuse across. All right, we have a similar sandwich here, formed of the simple squamous epithelium of the capsule, outer capsule. So this is the parietal layer, the outer layer. And then the inner layer, the visceral layer, as I indicated, was thrown into these folds where it wrapped around the capillaries. So if I bring in the afferent arteriole, here's the efferent arteriole leaving. You can always tell because the efferent arteriole in the models is more narrow and the afferent arteriole is wider. Right? That helps to increase the flow rate and the filtration pressure. And then we'll draw our glomerular capillaries. So when the plasma is filtered across our filtration membrane, now that it's in the capsule itself, we call it filtrate. And we don't call it urine again until it drifts into the minor calyx. Right? So all the time it's passing through the nephron, all the time it's passing through the collecting duct, we call it filtrate. It's being modified, so it's not yet urine, and doesn't have the same content as blood. So this region right here, the function of the capsule is filtration. So we have to have some small sizes. The cells of the visceral layer that form the filtration membrane are really weird-looking simple squamous cells. I'm going to draw this temporarily and then erase it. There's a wide body, and extending from that body are processes they look really weird. And so that would be one cell known as a podocyte. Again, that's the visceral layer of the capsule. Here would be another podocyte. Its processes would interdigitate. And there's a narrow space between each of those processes. That's part of the filtration. That's the most narrow of all the spaces that the fluid and its contents have to pass through. All right? Now if you look at that in an electron microscope view, instead of seeing what I have drawn up there, what we see instead, because it's a much thinner section, is we see the podocyte. Here would be processes coming down like this. And these foot-like processes are known as a pedicel. It interdigitates with another podocyte. Every other process belongs to this other podocyte. So this is the smallest opening that the glucose and amino acids have to pass through. And that gap, I'll draw it a little bit larger here. That gap is known as there's a little bit of a proteinaceous membrane. It's known as a filtration slit. So that's the fancy appearance of our visceral layer. Then we have our simple squamous of our blood vessel. So here's the blood in the glomerular capillaries. And here's the simple squamous epithelial cell. But it has pores. Like somebody took a paper punch and punched holes through the plasma membrane of the cell, top to bottom, and those holes seal themselves off. And so these holes, this would be the simple squamous epithelial cell. This would be our capillary wall. And these holes are known as fenestra. Or pores. And remember, every epithelium has a basement membrane. So also helping to determine size and charge is the basement membrane. So blood is on this side. What do we have on this side? Filtrate. Okay? And then out here would be the parietal layer. This is the capsular space. So I've essentially drawn one of these segments here. There. So blood is filtered out and the components are in the capsular space. Just to give you a quick view, all right, so here's a light microscopy. Here would be the glomerulus and the visceral layer. Here's the parietal layer out here. Here's the scanning electron micrograph of the podocytes that I drew on the board. What we can't see inside that wrapping would be the glomerular capillary. All right, and then here would be a scanning electron micrograph. So here would be the podocyte. And here would be the pedicels, shown down here. So every other pedicel belongs to a different podocyte, and down here are the fenestrae or pores. Filtrate up here, blood down here. Okay? And then we have that filtration occurring there. So here's another view. These are the pedicels. This is the plasma of blood. Filtrate, plasma. Okay. So now, I kind of illustrate this a little bit. When my kids, well, they still are not the neatest people in the world. When they were little and they were playing with their toys, I hated Legos. They were a pain to keep up off the floor, and my sister-in-law gave me about 500 pieces of Legos for the kids. And when I cleaned up the house, I had boxes, they were sorted out, but I just threw them away, rather than put them, because they were just going to end up on the floor. And I figured in 5 years I'd have half as many Legos to clean up as I started out with. The kids have their bedrooms off a hallway. My husband calls it the Hall of Weasels. And they walk down there and they drop a pencil, they drop a sock, whatever. If they don't need it, it stays there. They would walk back and forth over it for weeks. So let's say that I'm tired of cleaning up the house over and over and over, and I get a big box. And I walk through the house, and everything that fits into the box, I put into the box. And I take it out to the garage. That would be this process of filtration. Okay? And then the kids come home, and stuff that they haven't used for 6 weeks, all of a sudden they have to have. And they asked me where it is. And I said, well, I'm about ready to throw it away. It's in the box out in the garage. So they go out there and they take back from that box the stuff that they think they really need. Reabsorption. Okay? In the meantime, it's another week or so before I can take the box to the trash. I keep finding stuff around the house that I then take out to the garage and add back to the box. Secretion. Okay, and that's essentially the process that occurs in the kidney. So after we create the filtrate, the next thing that has to happen is reabsorption and a secretion, as you saw on the template. That, for the most part, is going to take place in the proximal convoluted tubule. We're not going to look at the pumps and the channels and sodium, secondary active transport, but I do want to look a little bit at the epithelium. So our next structure here is known as the proximal convoluted tubule. Proximal because it's closest to the point of origin. And we need a lot of surface area. So what structures do we have for surface area? Cilia or microvilli? Microvilli. So a cell of the proximal convoluted tubule is simple cuboidal. It has highly folded basal and lateral membranes and very tall microvilli. They're so tall they look like cilia. But they are non-modal. So these are microvilli. Increases the surface area. We have sodium potassium pumps down here transporting the salt out and creating a drag that pulls the glucose and amino acids out. So what organelles are we going to have lots of down here in the mitochondria? So we have, it's like they're stuffed. We have tons and tons, well not literally, of mitochondria in the bottom part of these cells. So that they can, this is facing the lumen, so the filtrate is going to be here. And the blood is going to be out here. So that provides the energy and the surface area for transporting material out of the proximal convoluted tubule. Okay? Then, the next structure is that nephron loop. May I erase the cell? The nephron loop has a thick section, but then it has a thin section. And I'm not drawing this anatomically because it actually should, well, maybe I'll swing it around. It actually should swing back this direction. And our nephron loop is formed of simple squamous epithelia, the thin segment. And then really the only nephron loop that we care about is the juxtamedullary nephron loop. Right? Because that's where it's moving sodium out into the interstitial fluid, and water doesn't follow. So the descending limb is permeable to water, but the ascending limb is not. Concentration is about 300 milliosmoles in the filtrate going down and the filtrate leaving. It's 100. You don't have to remember these numbers. It's not a physiology class. I'm just illustrating the filtrate is definitely not concentrated. It's actually become more dilute. And the reason for that is it's giving up salt. And so these particles become more and more and more concentrated the closer we get to the tip of our papillae. Those particles are coming out of the filtrate, hence it's much more dilute. Then we get to our distal tubule, and the distal tubule comes back and passes right by and in between our afferent and efferent arterioles. This is necessary, well, it's a role of sensitivity that allows the kidney to auto-regulate filtration pressure. So if filtration pressure is too low, then that is detected in the distal convoluted tubule. And so we have this relationship. Here's a cross-section of the distal convoluted tubule, where it's passing by afferent arteriole and efferent arteriole. And this is a unique relationship of structures known as the juxta, meaning next to, glomerular. So next to the glomerulus, I'm drawing a large view of this, apparatus. The cells of the distal tubule that face the glomerulus are very tall instead of cuboidal. And the nuclei become very close together. They're sensitive to the sodium content of the glomerulus. So these cells are known as the dense spot or macula densa. When they sense not enough sodium in the distal convoluted tubule, they release chemicals that affect the diameter of the afferent arteriole. In addition, there are smooth muscle cells in the afferent arteriole that are sensitive to stretching and collapse of the afferent arteriole. These are specialized smooth muscle cells known as granular cells. That's a newer name for them. Your lab manual, I believe, calls them juxtaglomerular cells, but that's too easily confused with juxtaglomerular apparatus. Okay, so granular cells or juxtaglomerular cells, JG cells, are the same thing. These are granules of an enzyme known as renin, and that's involved in a complicated sequence of activation of hormones that increase blood pressure in the body and increases filtration. But we have this specialized region known as the juxtaglomerular apparatus. So let me just show that to you, and because we have this on our model. So here's our capsule, the parietal layer and the visceral layer and the podocytes around the capillaries. Here's the afferent arteriole coming in, and we have the juxtaglomerular or granular cells. Here's the efferent arteriole, and here's part of the distal tubule just before it becomes convoluted. And so the distal tubule cells are a flat cuboidal epithelium. I'll diagram them on the board in just one moment. But here where they're taller and skinnier is the macula densa. So just that part of the tubule facing the capsule. Okay. And so the juxtaglomerular apparatus is functional in control of blood pressure within the capsule. Because when blood pressure is too low, we don't do filtration. When blood pressure is too high, it can damage the filtration membrane and spread those slits too far apart. All right, so that brings us then to the distal convoluted tubule. And the distal convoluted tubule has its cells are cuboidal, but they are of a flat cuboidal and very little microvilli. Because we've already taken back most of the material, okay? And we're not going to do very much. It's limited under the regulation of hormones. So that's the end of the nephron. The next structure, the collecting duct, all right, is going to start out in the cortex and pass into the pyramid. Several collecting ducts will come together to form wider papillary ducts, and they empty through the papilla. So the collecting ducts are very similar to the distal tubules. They're of a flatter cuboidal, but as they get close to the papillary ducts, they become a tall columnar epithelium. So the entire collecting duct or papillary duct is more distinctive. It's larger in diameter, and whether it's cuboidal or simple columnar, the membranes separating each cell are very distinct. In the proximal and distal tubules, I've drawn those membranes in there, but because they interdigitate, it's kind of wavy like this, it's hard to see those plasma membranes. So it's not as easy to identify the collecting duct. What I want you to remember about the collecting duct is in the medulla, as the filtrate passes through the collecting duct, this is the site where we determine whether or not the filtrate is going to become concentrated. So if you drank 10 glasses of water this morning before you got up, you're not going to be making ADH hormone. Aquaporins will not be in your collecting duct. The filtrate water will stay in the collecting duct and will enter into the minor calyces as a very dilute, pale-colored urine. If you haven't drunk any water since noon yesterday, your brain is going to be making ADH. Lots of water channels will be placed in the collecting duct. You'll take out lots of water to keep in your body. You'll have a scant amount of urine. It will be a dark yellow-brown color. It'll sink to the bottom of the toilet bowl. We have so much salts and urea and stuff in it. All right, so that's where it's in the collecting duct itself that we take advantage of the gradient of particles created by the nephron loop. Now, I know I did a lot of physiology in here, okay? If you look at the sample exam, the questions on that are more general. I'm not looking at the cellular processes, but essentially just kind of the basic function of each of these regions of the nephron, okay? All right, we didn't get to the urinary bladder. We'll look at those structures in lab. Okay, there's about 3 or 4 parts of the urinary bladder identified.